Imagine walking into a neurology clinic. You look across the waiting room and you see two patients sitting side by side.
Right, and on the surface, their charts look totally identical.
Exactly. I mean, they are both experiencing the exact same devastating symptoms. You know, the progressive loss of motor control, the muscle weakness, the fading ability to speak.
Right, they both have amyotrophic lateral sclerosis, or ALS.
Yeah, but if you were to somehow zoom in, like past their symptoms, past their tissues, and deep inside their spinal cords, you would see a massive biological paradox. At the microscopic level, their cellular immune responses are literal opposites.
It is just a stunning realization. We are talking about two entirely different diseases wearing the exact same clinical trench coat.
Oh, wow, yeah. That is a great way to put it.
And uncovering that difference fundamentally rewrites everything we thought we knew about treating neurodegeneration.
So, welcome to the deep dive. Our mission today is to unpack a groundbreaking 2025 paper published in Nature Neuroscience.
It is a phenomenal paper.
It really is. We are exploring how a very specific genetic stutter, a mutation in a gene called C9orf72, causes the brain's immune system to act in the exact opposite way we previously assumed.
Because for decades, the focus in ALS research has been almost exclusively on the motor neurons themselves.
Right, the cells that actually control movement.
Exactly. They are the wires connecting your brain to your muscles, and in ALS, those wires die. But this research forces us to pivot.
Okay, let's unpack this, because the motor neurons might be the primary victims, but the architects of the destruction, or at least the accomplices, right, are the support cells surrounding them.
Yes. So in a healthy person, this C9orf72 gene has a tiny sequence of DNA. It's the letters G, 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 C, C.
And that's totally normal to have, right?
Completely normal. It naturally repeats maybe a few dozen times. It is just a normal part of the genetic code. But in a subset of patients, those with what we call C9ALS, that sequence breaks.
It stutters.
Exactly. It expands massively, repeating anywhere from 38 to over 1,600 times.
Over 1,600 times. That is a massive glitch.
It is. And that massive expansion completely breaks the gene's ability to function normally.
So in biology, genetic mutations generally fall into one of two categories, right? You have your toxic gain of function, where a mutated gene starts producing something actively harmful.
Like a factory suddenly churning out poison.
Yeah, exactly. And then you have a loss of function, where the gene just stops doing its day job altogether.
And the researchers in this paper discovered that this specific stutter causes something called haploinsufficiency.
Haploinsufficiency. Let's define that for everyone listening.
So it means that because one copy of the gene is mangled by those repeats, it is essentially inactivated. The cell is suddenly operating on half power.
It simply doesn't have enough of the protein it needs to run its daily operations.
Right. But to figure out exactly what that power failure does to the brain, the researchers had to read the private diaries of over 150,000 individual cells.
Which is just an incredible feat of modern technology. I mean, they used single-nuclei RNA sequencing, right? RNA-seq.
They did, because instead of taking a chunk of spinal cord tissue and grinding it up into a biological smoothie to see what genes are active overall.
Which is how we used to do it.
Right, exactly. Okay. But this new technique lets researchers isolate individual cell nuclei. They looked at 156,252 nuclei from the motor cortex and the spinal cords of patients.
And they compared sporadic ALS patients, those without a clear genetic cause, alongside patients with the C9 mutation, and healthy controls.
Yes, so they can see exactly which genes are turned on or off in every single cell type at that exact moment in time.
And when they isolated those cells, the biggest shock wasn't what was happening in the motor neurons at all. It was what was happening in the microglia.
The microglia, which are absolutely fascinating cells.
Yeah, so if you listening are not deeply familiar with neuroanatomy, think of microglia as the brain's neighborhood watch. They are the resident immune sentinels.
They constantly patrol the neural tissue, looking for invading pathogens, dead cells, or toxic protein buildup.
Right, their job is to react. When they sense danger, they transition out of their resting homeostatic state and they armor up.
They turn into what we call a reactive disease-associated state, or DAM, and they categorize these into 6 microglial states, MG1 through MG6.
And in the single nuclei data from the sporadic ALS patients, that is exactly what researchers saw, right?
Precisely. The microglia were sounding a massive chemical alarm. They were upregulating inflammatory genes like FDH1, CD14, and HLA markers to fight the disease.
But the C9 ALS patients showed the exact opposite. Their microglia were completely suppressed.
Yes, they were expressing high levels of genes like P2RY12, which basically broadcasts everything is fine, we are just maintaining the status quo.
While the motor neurons right next to them were actively dying.
It's wild. They were just stuck in that homeostatic state.
So, to use our neighborhood watch analogy, in sporadic ALS, the microglia see a fire, they put on riot gear, grab the hoses, and attack the problem. But in C9 ALS, because of that haploinsufficiency, it's like they've lost their walkie-talkies. They are just sitting on the porch in their pajamas while the entire house brains down to the ground.
The contrast is just jarring. You have a hyperreactive immune response in one patient and absolute immune paralysis in the other.
But, wait, I have to pause and look at this skeptically for a second. If I'm looking at a diseased spinal cord, it's a mess of inflammation and dying cells.
It is a highly toxic environment, yes.
So how do we know it's the internal genetic mutation causing those microglia to freeze up, and not just something else in the patient's spinal cord?
That is a very fair point.
Like, couldn't the toxic environment just be suppressing them? Maybe the neighborhood is so full of smoke they just can't see the fire.
That is the exact hurdle the researchers had to clear next. They needed to prove it was an internal hardware failure, not an environmental side effect.
Okay, so let's do a quick 5-minute recap for you listening, just to lock this in. Bullet 1. ALS has sporadic and genetic forms, specifically this C9orf72 mutation.
All right, and bullet 2. Sporadic ALS microglia become hyperreactive. They armor up.
And bullet 3. C9 ALS microglia suffer haploinsufficiency and completely fail to react. They stay in their pajamas.
Perfect summary.
So here's a question for you, the listener. If you were designing a drug for ALS right now, would you want it to suppress the immune system or stimulate it? Keep that in mind as we go deeper.
That is the million-dollar question. And to answer your previous pushback about the environment versus the genetics, the team designed an incredibly elegant experiment.
Oh, this part is almost sci-fi.
It really is. They used a human brain cellular xenograft model, or HBCX. They took skin cells from a human patient with a C9HRE mutation and reversed their biological clock, turning them into induced pluripotent stem cells.
So they have these blank slate human cells carrying the genetic stutter?
Yes. And then they used CRISPR gene editing technology to split those cells into different groups.
Okay, what were the groups?
In one group, they kept the mutation. In another, they carefully snipped out the stutter, creating a perfectly healthy, corrected cell line called C9-ISO.
Isogenic control, right.
Exactly. And in the third group, they completely deleted the C9orf72 gene altogether. That's the C9 knockout, or C9KO.
So we have the diseased human cells, the artificially cured human cells, and they knocked out human cells.
Now for the wild part. They coaxed all those stem cells into becoming microglial precursors, and they injected them directly into the brains of living mice.
Wait, I want to make sure we visualize this properly. We have human immune cells living, growing, and patrolling inside a mouse's brain.
Exactly. And they didn't use just any mice. They used a highly specialized immunodeficient strain called, and get ready for an acronym, RAG2 IL2G HCSF1KY.
Okay, yeah, that is a mouthful. Unpack that for us.
Basically, the RAG2 and LTG knockouts mean the mouse has absolutely no immune system of its own. No T cells, no B cells, no natural killer cells.
Ensuring the mouse wouldn't reject the human cells.
Right. And the last part, the HCSF1KI, means they engineered the mouse to produce a specific human survival protein. So the human microglia feel right at home.
That is brilliant. So they let these human cells live in the healthy mouse brain for months. They completely removed them from the toxic dying environment of the human ALS spinal cord.
Yes.
So if the environment was the problem, these cells should wake up and act normal in a healthy brain.
But what's fascinating here is they didn't. When the researchers extracted those cells months later using FACS fluorescence-activated cell sorting, the results were definitive.
FACS, where they use lasers to sort the cells one by one based on glowing markers, right?
Exactly. And the C9 mutated microglia still failed to activate. Even in a perfectly pristine mouse brain, they couldn't armor up. They failed to upregulate key reactive genes like HLA and SPP1.
Wow. And what about the knockout cells?
Vitally, the cells where the gene was completely deleted, the knockout cells behaved the exact same way.
Which proves it is entirely an internal hardware issue. It's a fundamental loss of function. The C9 mutation directly causes the immune paralysis.
Right. The cells aren't confused by the smoke. They genuinely lack the biological machinery to respond to the fire.
And this immune paralysis actually mirrors another devastating disease, doesn't it?
It does. The researchers compared this to GRN-FTD, which is progranulin deficient frontotemporal dementia.
So a type of dementia, not a motor neuron disease.
Right. But surprisingly, the C9 ALS microglia transcriptomes positively correlate with GRN-FTD. They share massive molecular similarities.
Whereas the sporadic ALS microglia negatively correlate with it.
Exactly. It reveals how these neurodegenerative diseases might be categorized less by the symptoms they cause and more by the cellular machinery that breaks down to cause them.
So synthesizing where we are, let's do another quick 5-minute recap.
Yeah.
Bullet 1. Human microglia grown in mouse brains prove the C9 mutation directly impairs activation.
Bullet 2. The C9 knockout cells mirror the disease cells, proving it's a loss-of-function issue.
And bullet 3. C9 ALS shares molecular similarities with certain types of dementia, like GRN-FTD.
Spot on.
So, question for you listening. We've proven the immune cells are stuck, but why are they stuck? What machinery inside the cell is physically broken? Let's zoom in.
To find that broken machinery, the team deployed an algorithm called WGCNA, or Weighted Gene Co-expression Network Analysis.
Okay, that sounds intense. What does it actually do?
It basically finds biological systems that are failing together. And by analyzing the data, WGCNA pointed a massive red arrow at one specific internal system, the endolysosomal pathway.
The endolysosomal pathway. Okay, let's translate that out of textbook speak. This is basically the cell's internal digestive tract, right? It's garbage disposal.
Yeah, that is a perfect way to look at it. When a microglial cell is patrolling and finds toxic debris, say a clump of dead proteins, it eats it.
It swallows the trash.
Right. It swallows it into a biological bubble called an early endosome. As that bubble moves deeper into the cell, it matures into a late endosome. Finally, it merges with the lysosome.
And the lysosome is essentially a vat of acid?
Precisely. It is highly acidic and packed with digestive enzymes, most notably one called cathepsin D, or CTSD. The lysosome breaks down the trash into basic components so the cell can recycle it.
So to see if this garbage disposal was physically jammed, the researchers used a technique called expansion microscopy alongside transmission electron microscopy.
Yeah.
And expansion microscopy is fascinating. They literally inflate the tissue with a polymer, like a water balloon, so they can see the tiny internal structures under the microscope with much greater clarity.
It's an incredible visualization tool. And what they saw inside these cells was a massive microscopic traffic jam.
What did it look like?
Well, the C9 mutated cells had these hugely enlarged lysosomes hoarding that cathepsin D enzyme. And even worse, they had a massive buildup of late endosomes.
The holding tanks for the trash.
Exactly. And we know they were late endosomes because they tested positive for INP1 and REB7, which are markers for the late stage, but negative for EEA1, which is the early endosome marker.
So seeing a traffic jam is one thing, but proving the traffic isn't moving is another. This is where they ran what I think is the most brilliant functional test in the entire paper.
The phagocytosis experiment.
Yes. Here's where it gets really interesting. They wanted to watch the garbage disposal run in real time. So they fed the microglia E. coli bacteria.
But not just normal bacteria. They used E. coli photobioparticles.
Right, which act like molecular glow sticks.
Yes, the photo dye is sensitive to acidity. So these bacteria only glow bright fluorescent green when they reach the highly acidic stomach of a functioning lysosome.
It is a phenomenal way to track digestion. If the cell just swallows the bacteria, nothing happens. But if the cell actually digests it, it lights up green.
And the timeline here is what makes the findings so profound. After 30 minutes, they checked the cells.
And what do they see?
The healthy control microglia and the C9 mutated microglia looked identical. They had both swallowed the bacteria perfectly fine. Their initial uptake was completely normal.
So shoving the food down the drain worked flawlessly. But then they left the cameras rolling for 900 minutes.
Which is 15 hours.
A long time for cells.
Exactly. And 15 hours later, the healthy cells had done their job. They had broken down the bacteria, and the green glow was fading away.
But the C9 mutated cells were still glowing bright green.
They were. The undigested bacteria were just sitting there. They completely failed to degrade the cargo compared to the healthy cells.
To build on our garbage disposal analogy, you shove the food down the drain perfectly fine, but the blades are jammed. You flip the switch, and the motor just hums. The trash just sits there piling up.
And because the blades are jammed, the sink starts overflowing. The cell is expending all of its energy just trying to manage this massive backlog of undigested late endosomes.
Wow.
A cell that is physically choking on its own garbage absolutely cannot transition into that active disease-fighting armor we talked about earlier.
Right, its internal resources are entirely compromised. So time for another quick recap. Bullet 1, the physical defect is in the endolysosomal pathway, the cell's digestive tract.
Bullet 2. Biomarkers show a massive buildup of late endosomes, which are LMP1 and RB7 positive.
And bullet 3, microglia can swallow debris perfectly fine, but cannot digest it, which we saw in the 900-minute Frodo experiment.
Exactly right.
So here's a question for you listening. If the microglia are hoarding all this undigested trash, how does that affect the other cells around them? Are the neighbors noticing?
And that brings us to another vital support cell, the astrocyte.
Astrocytes, I love these cells. They don't operate in a vacuum, right?
No, they don't. Astrocytes are these incredible star-shaped cells that wrap around synapses, regulate blood flow, and essentially manage the local environment of the brain.
They're like the scaffolding and the paramedics combined. And normally, if the motor neurons are dying, you would expect the astrocytes to be mounting a massive emergency response alongside the microglia.
You would. But the SNRNA-seq data showed another shock. In the C9 ALS patients, the astrocytes were also asleep.
Also asleep.
Yes. Just like the microglia, they had a vastly diminished reactive response compared to sporadic ALS. They were expressing homeostatic genes like AQP1 and SLC35F1.
Instead of disease-fighting ones. But wait, the C9orf72 mutation predominantly impacts the microglia, right? So why are the astrocytes also failing to react?
The researchers suspected a communication breakdown. Cells constantly talk to each other using chemical signals. One cell releases a ligand and another catches it with a receptor.
So to intercept these text messages, the team used an algorithm called cell chat, right?
Exactly, cell chat. It's a ligand receptor mapping tool. It basically maps out the group chat of the brain.
That's a great way to put it.
And the group chat in sporadic ALS look fundamentally different from C9 ALS.
Okay, let's look at the sporadic ALS chat first, where the immune system is hyperactive. What were they saying?
In sporadic ALS, cell chat found a massive engagement of a specific pathway called the SPP1-CD44 axis.
And SPP1 is basically a broadcast signal that screams attack.
Yeah,
right?
it is. It drives aggressive inflammation. The cells are constantly yelling at each other to escalate the fight.
But in the C9 ALS group chat, what's happening there?
Complete silence on the attack channel. Instead, there was an increase in a totally different pathway, the JAS6-AXL-MERTK axis.
And GS6 is definitely not an attack signal.
No, not at all. GS6 is a signal that literally binds to immune cells and tells them to stand down. It puts the brakes on the inflammatory response.
So what does this all mean? We have a chemical stand-down order happening in C9 ALS and a chemical attack order in LS.
It means the damaged microglia act as a suppressive anchor on the entire neural ecosystem. They're actively broadcasting a chemical sedative to the rest of the brain.
Forcing the astrocytes to stay asleep while the disease progresses?
Yes, it is a cascading failure.
So, final 5-minute recap for you. Bullet 1, astrocytes in C9 ALS also fail to activate.
Bullet 2, cell signaling is fundamentally altered between the two forms of the disease.
And bullet 3, the GS6-ASL axis puts the brakes on the immune response in C9 ALS.
Right.
So think about patient treatment, listener. If you give a general ALS patient an anti-inflammatory drug, what happens if they have the C9 mutation?
That is the grand, terrifying synthesis of this entire paper. The big takeaway.
Let's lay it out.
Sporadic ALS and C9 ALS might look clinically indistinguishable on the outside, but their cellular substrates are total opposites.
Right.
Sporadic ALS is characterized by hyperreactivity. C9 ALS is characterized by immune paralysis and a broken cellular digestive system.
So we absolutely cannot treat all ALS patients the same. Precision medicine and patient stratification are absolutely critical now.
They are no longer just hypothetical buzzwords. They are clinical necessities.
Because if you have a patient with sporadic ALS, their brain is on fire. A drug designed to cool that fire down to suppress the immune system might buy them time.
It makes logical sense for them, yes.
But if you take that exact same standard anti-inflammatory treatment and give it to a C9 ALS patient,
you are sedating an immune system that is already asleep on the porch.
the GI6 pathway is already forcing their astrocytes to stand down.
Their microglia are already paralyzed, choking on undigested endosomes.
So if C9orf72 mutations cause an underactive microglial response, it raises a terrifying but vital question to leave you with. Could standard anti-inflammatory treatments drugs designed to calm the brain down actually accelerate the disease in these specific patients?
It's a very real possibility.
It forces us to ask, as we design the future of neuromedicine, are we treating the symptoms we see or the cells we can't?
That is exactly what we have to figure out next.
Thank you for taking this deep dive with us.